Hello everyone. I am Chayanon Kitkana from Faculty of Medicine, Konkan University, and today I'm going to talk about the work from our research group that study about the biomarker of meningioma recurrence. So let's start with a bit of background about why we choose to study the meningioma recurrence. Meningiomas are common CNS tumor, often benign but potentially dangerous due to their location. Recurrence is also possible despite the surgical treatment and requiring a long-term monitoring. Current recurrent risk assessment rely heavily on WHO grading and also clinical and histopathological data, which have limitations in accurately predicting the tumor recurrence. And studying the molecular biomarker can enable more precise and early recurrence prediction, enhancing patient management and outcome. And uh, in this work, we address a small dataset problem by combining a microarray with sequencing data. We use a length in a work by Tang et al. In 2021. Uh, we retrieve our gene expression data from gene expression omnibus, and then we divide it into two cohorts, a validation cohort and a discovery cohort. Each cohort have one sequencing data and one microarray data. We use LangIn to perform a weighted length transformation to account for the difference in the distribution between the sequencing data and the microarray data. And we also use LangIn to perform the differential gene expression analysis, then validated the differentially expressed gene using the discovery cohort. And from the pipeline, the output is the validated differentially expressed gene. We further define the list of genes by trying to reduce the multicollinearity in the data by using the variant inflation factor. We recursively removing the gene with the highest variant inflation factor until there is no gene with the variant inflation factor more than 3 left. Then we use the output from the previous pipeline to perform the model selection. And in the end, we choose the logistic regression model for its interpretability and its performance. And these are the top 5 genes that we found. Our model used 41 genes as an input. And to wrap up our summary, we identify a new set of genes that can predict the meningioma recurrence with a better performance compared to the previous work. But there is some limitation about our work. Recursively removing the gene with the highest variant inflation factor may lead to arbitrary gene selection or introduce bias in the analysis. So we recommend integrating the biological knowledge into performing the feature selection. And also, this study only made a classifier, so a time to recurrent analysis of this candidate will further enhance their utility. Thank you for listening.
So do you have questions? Please feel free to ask.
Yeah. So your model is reaching 1. Is it overfitting?
We did validate it with external external validation. It's a data set from another cohort that is independent from the discovery cohort.
Oh, that's good.
Okay.
It means it's good.
So we have one question from virtual participant. So can you explain the calculation head VIF?
VIF. Basically, is fitting the linear regression model. Or for example, if I have 5 genes A, B, C, D, E, and I want to find the variant inflation factor of E, I'm fitting a linear regression model to predict the expression of E based on the rest of the genes.
So you're taking a panel of genes basically.
Yes.